All right, great to see you guys. Hope you're having a good week so far. Wonderful. All right, we'll wait just another minute here. Anybody have any questions? All right, Abigail.
So I have a question. Just to let you know, I can't do the experiment today because my bromothal blue didn't come in time, but I will be able to do it just later.
Okay, that's fine. You have the instruction sheet, the lab sheet that I gave you. You can use all the instructions on there. And watching today will be helpful so that when you do it yourself, you are, kind of know what you're doing. So another thing, those of you who are not able to participate today, we will be using that bromothymol blue at least one more time, maybe twice over the course of the year. So if you can get it and be ready for the next time, that would be great. If not, I do understand it's not always possible. Some of you guys live in places where it's not easy for you to get supplies. So if you're not able to, you can observe those. I always make it real clear when something is required. Whether you do the experiment or not today, though, you do have to complete the lab sheet because we will talk about it next week in class. So make sure that you complete it and bring it completed to class next week. All right. If nobody has any other questions, I will, there's a chat here. So the question is, will we still be doing Pivot Interactives? I'm trying to find a Pivot Interactive that I think is a good one. I actually think Pivot Interactives are a really awesome resource. So if you guys ever want to go do them on your own, I think that is a good idea to do them on your own. As far as doing them together or me assigning them, the problem with them is they take a long time to do. And I know that time is an issue for a lot of you guys. A lot of the reason that your families choose homeschooling is so that you can not have to spend a ton of time doing a lot of busy work. So I'm working on it. I'm working on a way to sort of cut out a lot of the extra work that's in there. So hopefully we will, hopefully. All right. So hopefully you can see that where it says biology is life. One little bit of information before we go on. You guys are doing a great job participating. But I did have someone tell me that this person was raising their hand sometimes and I wasn't seeing the hand. I wasn't responding. And they were asking, can you see it when I do that? Well, sometimes yes, sometimes no. One of the problems is that if you hold your hand up like this, if like that's how you're trying to let me know that you want to answer, I may just not see that. And sometimes that little yellow hand that goes up in the corner there, the little virtual hand, sometimes it's just not very visible depending on what the background is. Like as an example, if you look at Abigail and Christopher's pictures there, you can see where there's the little bit of white, like from the window. If that hand is on top of that, I might not see it. Yeah, right there. It might not be visible. So if you want to answer and I'm not seeing the hand that you put up, just try moving it. Try moving it around a little bit. One other possibility is that we all have different internet speeds. So it's possible that your hand, either virtual or real, isn't going up until after I've already called on somebody else because the timing is off. So I'm not getting it until after you're done. So if you're not being called on, move your hand a little bit or type a question mark in the chat box. That lets me know that you want to answer. I'm not ignoring anybody. So I do my best to try to call on everybody. And I want to just encourage you again to continue participating. I think you learn a lot more when you do rather than just watching. Okay. All right. Did any of you guys see the Northern Lights, the Aurora Borealis a couple of days ago that were it's been about 5 days ago now. So anybody who lives in the northern part of the United States, from like the Midwest over to the east, all the way down to the Carolinas, they were showing the Northern Lights. And I just wondered if anybody had had a chance to see them. So Northern Lights are where there's these bright, vivid, beautiful colors in the sky. It's caused by a solar storm when you've got a lot of energy, a lot of plasma is coming from the sun, and it makes its way to our Earth's magnetic field that we have around the Earth that protects us from the harmful rays of the sun. Well, sometimes there's a lot and it causes a reaction up in our atmosphere that gives us these beautiful, beautiful colors. But I didn't see them, even though I went outside like every 15 minutes for hours. Both of the nights I didn't see them. So I saw this cartoon that I thought was pretty funny. I didn't see the Northern Lights. I just like to be included. So somebody drew with markers. Yeah, that's what it felt like. It was like, darn, I didn't see them and I wanted to. All right. So let's talk a little bit about this egg lab, the osmosis lab that you guys had as an extra credit lesson. Now, not many of you did that. I do recommend that you try to do as many of these labs as you can. Not only do you get extra credit when you do some of these labs, but you really do learn a lot. And this is kind of an important one if you can. And it's just really cool. So to go back and do it even now is not a bad idea. All right, so I want to talk a little bit about this lab for those of you who did it. Oh, there it is, Christopher. Look at that. Yeah, look at the size of that egg. Wow, that's a big one. So that must be in the vinegar, right? Yeah. Yeah, so we'll talk about each step of the way. So eggs, think about what an egg is. An egg is a cell. Pretty crazy to think of that. An egg is the largest cell that most organisms make. The reason for that is because the egg has to contain almost everything that the organism needs. The egg cells have all the organelles in them. They have all the energy sources. They have the cell parts that are needed for development of the new organism. The only thing that sperm contributes in fertilization is DNA. It contributes genetic material, and that's it. Everything else is inside the egg. So it's pretty big. Now, bird eggs are some of the largest single cells that we have on Earth. And once they're fertilized, they begin growing really quickly. Billions of cells eventually form into those tissues and organs and organ systems before the bird eventually hatches. Now, we did use a chicken egg in this lab to allow us to have a model to see how the inside of the egg is affected by the environment on the outside. So the very first thing that we did is put that egg in a solution of vinegar. That's what Chris's looks like. Why did we do that? What was the point of putting that egg in vinegar? Hopefully, even if you didn't do the lab, you read about it, so you should know the answers to these questions. Why did we put the egg down in vinegar? For what purpose? This is where the class participation part comes in. All right. Now, you're on the right track, but watch your wording. Degrade isn't exactly the right word. But you're on the right track, Chris. Bethany.
I believe I've done this experiment before. If my memory is correct, it's to see the inside of the egg.
Yes, but what does it do to it that allows you to see inside?
Something to the outer shell. I can't remember.
Yeah, you're right. You're right. You guys are walking around it. They dissolve the shell. They break it down. So chicken eggs, most eggs that have a hard shell are made out of calcium carbonate, which is the same thing that shells are made of. Seashells are made of calcium carbonate. Now, that calcium carbonate reacts with the shell and breaks it down and dissolves it. Once that shell is dissolved away, what did we see happen? I mean, we just saw it in Chris's egg. Once the shell was gone, what happened to the egg?
The egg becomes see-through.
Becomes see-through. It becomes translucent. And what else? I made a comment about that egg when Chris held it up.
It grows.
Yeah, it gets really big. Now, it gets larger. Anybody have any idea why it got larger? It's not alive, so it's not really growing, but it certainly is increasing in size.
Maybe because the eggshell was stopping the egg from growing larger?
No, that's a good thought, though. Yeah, these eggs are not alive anymore. The eggs that we used, and certainly once you dissolve away the shell, there's no possibility of fertilization anymore. So there's no life inside there. But it did something to it. Dissolving that egg did something. Bethany.
Did it absorb some of it?
Yeah, it's going to absorb something. Yeah. So what's it sitting in? What's that egg sitting in that dissolves it away?
Vinegar.
Vinegar, and vinegar is mostly what? Vinegar is mostly water. There's a little bit of acetic acid in it, but it's mostly water. So what happened was once we took that outer shell off, now the inner membranes are semi-permeable. They let things move. Well, there is so much, so high a concentration of particles inside the egg that the water and the vinegar move through the membrane to get into the egg to spread those particles out. I know none of us like the term social distancing anymore. We all kind of shiver when we hear that, but that's kind of what water does. Water is the social distancing police trying to spread those particles out. You can't be too close together. I got to move you apart. Tries to make the concentration of particles the same on both sides of the membrane. So once that shell was gone, water moved through that membrane into the egg through osmosis. So then the next step of the egg experiment is we pour out the vinegar and we cover the egg with Karo syrup. What happens when you cover it with Karo syrup? Anybody gotten that far? The egg is going to shrink when you put it in Karo syrup because of osmosis in the opposite direction. So the corn syrup is mostly sugar. Now the water is going to leave the egg and try and spread out the sugar particles that are in the Karo syrup. It has a lower concentration of water inside the egg than on the outside of the egg. So the water is going to move out. It's going to try to reach equilibrium through this process of osmosis. Excellent. Now, I had a couple students write to me. I don't think it was anybody in this class. I think it was the other section, that the shell didn't seem to be dissolving. So they put the shell down in water and it didn't seem to be dissolving. And all of the non-dissolving eggshells were made from brown eggs. I don't know what a Malaysian red plant is. If it grows fully underwater, that should work. Yep, that should be fine. So, well, anyway, all of the non-dissolving shells were brown eggs. So I decided to run an experiment on my own to see if there was a difference in the way that brown and white eggshells behave in this experiment. So why were the students saying that the brown eggs weren't working? Now, this is what the eggs look like at the very beginning. Blue eggs, yeah. Eggs come in different colors depending on the breed of the chicken. That's why brown eggs come from chickens that have brown feathers and white eggs come from chicken that have white feathers. Chris, do you know what kind of chicken? You have your own chicken coop? That's cool. Do you know what kind of chicken is that lays blue eggs? You're muted. You're muted. We can't hear you. Hit your space bar.
Sorry, I was asking my mom. Hold on.
Oh. All right, so this is what my eggs looked like at the very beginning before they were placed into the vinegar. So notice here how you can see the spoon and notice about the approximate size. So I put them down into vinegar and I immediately started seeing bubbles from this. Now, the reason that vinegar dissolves the eggshells is because it has that acetic acid in it. That acetic acid reacts with calcium carbonate in the shell. Chris?
Americanas.
Americanas. Are all their eggs blue?
Most of them, but some are white, but most of them are blue.
That's pretty cool. Yeah. Yeah, you can get eggs that are in the pinkish range. Sometimes the eggs have a yellow shell on them. So that's pretty cool. All right, so here's how this reaction that we're seeing right here, this is how the reaction works. The acetic acid in the vinegar reacts with the calcium carbonate in the shell and it produces these bubbles. Anybody take a guess what these bubbles are made of? What's coming off?
Acetic acid.
Acetic acid is in the vinegar at the beginning, but it's having a chemical reaction. Remember what a chemical reaction is. All of the atoms are taken apart and they're rearranged and put back together to make new products. Bethany?
Water?
It is not water. That's a good guess, though. There's water that is produced, but that's not what the bubbles are. Water goes out into the solution right here, the vinegar solution. Maria?
Is it carbon dioxide?
It is carbon dioxide. Yeah. Carbon dioxide gas is one of the products of this reaction. So we get what's called calcium acetate. That's what's kind of making the water a little bit cloudy looking, and the carbon dioxide gas and the water. Now that carbon dioxide gas forms those bubbles, and you can see those bubbles going as the reaction occurs. All right. Now, once it had sat there for 2 days, I left it for 2 days in the vinegar. This is what happened. Now, they're both a lot bigger. So clearly something happened, but it doesn't look like the eggshell is gone. If we compare the two, this is what it looked like before. This is what it looked like after. This becomes very translucent, but this one still kept its brown color. So the reason is because the actual membranes inside the eggshell are brown. So that's why it doesn't look like they dissolved away, even though they did. Now, you can see here how big these eggs got. They're much bigger after they were in the vinegar. And then I poured out the vinegar. I put the Karo syrup on. I left it for a little more than a day. And this is what the eggs looked like. So here it shows the eggs. Look at how they're very soft. They're very squishy. They definitely shrank. A lot of that water came out. But a lot more water came out of the white egg than the brown egg. I wasn't completely sure why. So I did a little more research, and this is what I found. First of all, there were a couple of studies that I was able to find, and they compared white eggs and brown eggs. Now, eggs of the same size were used. That's important because if we start with one big egg and one small egg, we won't know. That's two variables now that we're changing. We only want to change one variable. So eggs that were brown weighed more than eggs that were white. Same size, more weight. Any idea why? Why would the brown ones be heavier than the white ones? The shells were thicker on the brown egg. A brown shell is a thicker shell. If you've ever cooked with brown eggs and white eggs, it takes a little bit, you have to hit the egg a little bit harder to break it, to crack it when it's a brown egg. Now, brown eggs also had a greater, what's called static compression shell strength. Anybody want to take that terminology apart and tell me what that means? Static compression shell strength. What do you think static compression shell strength is? Static means holding still. What's compression mean?
Like, not necessarily to squeeze, but to, I keep wanting to say compress, but like, I don't know how to explain it.
I know it's hard to not use the word compression when describing it. Yeah, but like downward pressure to the inside. Yeah, exactly. So it's basically, it's the ability of that shell to withstand that kind of pressure without cracking. And this is how they tested it. So eggshells are curved. They're very thin, but they're curved. And that curve helps them to distribute the load over the whole surface. So when you put compression down this way, that load is being moved all the way along. It's not just being carried right here. The load force is being moved all along the curved part of the egg. We actually use these curves in a lot of the things that we build. Buildings and airplanes and other engineering designs use curves. All right, so that's one thing. We found when we did this static compression shell strength test that the brown eggs were stronger. The brown eggs were able to stand up to that compression more than the white eggs were. Another thing that I found is that this membrane right here on the outside of the yolk, called a vitelline membrane, it is stronger than that of a white egg. So the brown egg has a stronger vitelline membrane. Now, for our lab that we did here, the vitelline membrane probably isn't very significant because we're concerned about the membranes that are on the outside. Those are the ones that we're wanting to make semi-permeable, that we're watching for. So when we're talking about those outer shell membranes, there's actually two of them. There's an inner one and an outer one that are along the outside of the egg white. They're right beneath the shell. They do a really good job protecting that egg. They keep bacteria from getting in. They allow oxygen and carbon dioxide to move across. Now, what's interesting is once that shell, which the shell is different, between a white and a brown egg. But once that shell was gone, was dissolved away, there was no difference between the membranes of a white egg and the membranes of a brown egg. We would expect there to be, because our eggs behaved a little bit differently. So for some reason, there was less water movement out of the egg with the brown egg. And I really couldn't find an answer for that. So my next step would be to run this experiment over and over. I only did it on 2 eggs, 1 brown, 1 white. That's not a good enough sample size. So in the future, I will tell everyone to use white eggs so that we all are comparing similar results with one another. All right, now we're going to move on to photosynthesis. So you guys have been learning about photosynthesis, and you're going to finish it and then start learning about cellular respiration soon. Hopefully you were able to get the supplies for today, but all you really have to have is the lab sheet that I sent you this morning. So I want to do just a little bit of review, some things that you need to know before we go into doing the lab. So this right here, there's some things that go into the reaction, and then there's some things that come out of the reaction. The reactants are the ones that go in. What are the reactants in, I don't have any today. I don't think I have any today. Yeah, I do if we get to them. I don't know if we'll get to them, but I might. All right, so what do we call, what are the reactants of the photosynthesis equation? What goes into a plant that it uses in order to perform photosynthesis? Think about what a plant takes in. Takes in something from the soil. What does it
Sunlight.
take? Sunlight? Okay, we'll come back to sunlight. You're right. It absolutely needs to happen in the reaction, but well, I won't come back to it. We'll talk about it now. Sunlight is absolutely necessary for that first phase, for the light reactions in photosynthesis. But when we talk about the reactants, we're talking about matter, things that have mass and take up space. Energy is different than matter. So sunlight is not really a reactant. You will, though, see it sometimes listed in the photosynthesis reaction. I have an example of that to show you here in a second. But you're right. We can't do photosynthesis can't happen without sunlight or some kind of light. That solar energy there. But the things that go in, what does a plant get from the soil? Chris,
Nutrients.
sorry, what?
Nutrients.
It does get nutrients. What does it get that it needs for photosynthesis? What goes in through the roots? Water, right? Plants absorb water through their roots. And then it gets something from the air. What do plants take in from the air? Same thing we breathe out. We breathe it out, they take it in. Maria.
Carbon dioxide.
Yeah, carbon dioxide. So there are our reactants. Now notice that solar energy is up here. You got to have solar energy in order for photosynthesis to happen. But what are the products? What are the things that are made in photosynthesis?
Sugars.
Yep, sugars in the form of glucose. And then one more thing, a waste product. Maria.
Oxygen.
Yep, oxygen. Those are our two products that come out. So this is the photosynthesis equation. Carbon dioxide plus water yields, that arrow means yields, glucose and oxygen in the presence of solar energy. All right, now, there are 2 stages of photosynthesis. Photosynthesis happens inside the chloroplast. It's that green plant organelle. What is the first stage of photosynthesis called? Anybody remember? There's a hint right here. It is called the light reaction. The light reaction requires light. This is where the need for sunlight comes in, is that first stage. The second stage of the photosynthesis is called the Calvin cycle, or the dark reaction, or the light independent reaction. They all mean the same thing. So take a look here at the light reaction. Now, ATP right here, that stores energy, and NADPH carries electrons. Now, since this phase needs light, it's called the light dependent reaction. So here come the water. Water came in through the roots, went to the plant cell, went into the chloroplast, is taken into the light reaction. But the only thing that's needed is the hydrogen. That oxygen is not needed. So the oxygen is released as a waste product. It's a lucky thing because organisms like us that need oxygen, we're able to breathe that oxygen in that plants put out there. That hydrogen in the light reaction is added to this NADP right there. Once the hydrogen's on it, it's called NADPH for hydrogen. This ADP, that's adenosine diphosphate, and a loose phosphate are added together to make ATP. Anybody remember what ATP stands for? ATP, ADP, important molecules to remember. ATP is adenosine triphosphate. It's an adenosine molecule attached to 3 phosphate molecules. ADP is adenosine diphosphate, 2. So when we break off one of those phosphates, lots and lots of energy is removed. All right, so in the light reaction, water goes in, oxygen comes out. NADPH and ATP move on to the dark reaction. All right. All right. Now, if you're ready, let's do our experiment to see how all this works. Now, get your lab sheet out. Whenever you have a lab sheet like this, you always want to read the whole thing. So we're going to read it together. What is the objective of this lab? What are we trying to do? We're trying to observe how plants utilize carbon dioxide. Remember, they take it in, and how they release oxygen during photosynthesis using bromothymol blue, a pH indicator to detect changes in carbon dioxide concentration. All right, here's your background information. Here's what you need to know. Bromothymol blue, that's what mine looks like right here, little jar. Bromothymol blue is a pH indicator that changes color depending on the amount of carbon dioxide present. In neutral conditions, the solution is blue. As carbon dioxide dissolves in water, it forms carbonic acid, which lowers the pH, turning the solution yellow or green. During photosynthesis, plants take in carbon dioxide from the environment to produce oxygen and glucose, which can cause a decrease in the amount of carbon dioxide in the surrounding solution. Okay, the term pH keeps coming up here. What's pH mean? What does pH even tell us? Anybody heard of pH before? Bethany.
Is it like the acidity of something? I feel
Yep.
like, yeah.
That's exactly what it is. It's how alkaline or how acidic it is. Sometimes we use the term base for alkaline. So it's either high pH, which is alkaline, or low pH, which is an acid. 7 is neutral. So right here, you can see the pH of different substances. So this would be neutral right here, plain water. We don't find pH 7 water on Earth because it's always mixed with something else. But in a lab, or the water that comes out of, like a purification system might have a pH of real close to 7. All of these things here are going to be a little bit more alkaline. All of these things here are going to be a little bit more basic as you go up or down the scale. Now, something has a high pH, or when it's basic, when it has alkaline in it. Something has a low one when it is acidic. Now, the reason that bases have a high pH is because they have more hydroxide ions. The more hydroxide ions you get, the higher the pH is going to be. Now, those hydroxide ions make it less acidic, so they're more basic. Something that has a low pH has a lot of hydrogen ions in it. The more hydrogen ions there are, the more acidic it's going to be. Now, I always remember the difference, whether there's hydrogen or hydroxide, by thinking of where hydrogen is on the periodic table. Hydrogen is real little. It's the smallest element. It only has 1 proton in it. one electron, one neutron. So hydrogen only has one. So I remember that the smallest pH numbers have more of the smallest element in them. The smaller the pH, the more of that little hydrogen that it has in it. Now, this is what the color of bromothymol blue looks like at different pHs. So we're going to take this, we're going to look at this when we do ours and figure out what the pH is when it starts and how much it is when we put the carbon dioxide in it. So I'll come back to this, but I just want to show you, we start here at pH 5.5. Remember, 7 is neutral, and then 8 is a little bit more basic or more alkaline. All right, Bethany. Were you
So
raising your-
we're looking at the pH of the stuff, the stuff.
You don't have to write the pH down. I'm just going to refer to it in a minute. I just wanted you to see this. Yeah. Okay, so the next thing we do is make sure we have all our materials. So I sent this out. Hopefully everybody has it. We've got the goggles, the bromothymol blue, the small water plant, 3 test tubes or jars, the beaker or measuring cup, water, a light source, measuring spoon or droppers, something to seal up your test tubes. I have little lids for my test tubes, so that's what I need. Aluminum foil, a drinking straw, and wet paper towels in case you get it on you. As we're doing this experiment, if you splash a little bit of the bromothymol blue on you, wipe it off with the paper towels. It's a little bit of an eye and a skin irritant. It's not going to burn you. It's not going to blind you, but you want to wash it off if you get it on you. All right. Now, the next thing is the hypothesis. So let's read that together. What do you think will happen to the color of the bromothymol blue solution when the plant undergoes photosynthesis in the light? What will happen in the dark? So when we're writing a hypothesis, it helps if we think of what is our independent variable? What is our dependent variable? So remember, the independent variable is the one you change. What are we changing in this lab? What are we trying to do? Look at that sentence there. What will happen to the color of the bromothymol blue when the plant undergoes photosynthesis in the light and in the dark?
The amount of light and the amount, like,
Yeah.
changing the light.
Yeah, exactly. The light. That could be one of our independent variables. Now, we're also putting a plant in, some of it, in one of the tubes, but not in another one. So the plant is actually a second variable. So this is not a controlled experiment that we're doing because we're going to change 2 variables at the same time. It's still valuable. They don't always have to be controlled in order to be valuable. So what is our dependent variable going to be? What do we measure? What are we going to be watching for?
The color of the bromothymol blue.
Excellent. Yep. So now that we know our independent variable, that's our if, and our dependent variable, that will be our then, we could come up with a hypothesis. If a plant is placed in bromothymol blue and placed in the light, then the solution will change color from yellowish green to blue, or from blue to yellowish green, or whatever we think is going to happen. All right, so that's a possible hypothesis for that one. All right, once you look at your procedure, whenever you have a lab that you're doing, always read the whole procedure first. First thing we do, put on our safety goggles. We're just going to read through it now, and then we'll do it together. Put 150 milliliters, which is a little more than a half a cup. A half a cup is 4 ounces, so we've got about 5 ounces. The amount of water is a little bit variable. Put that 5 ounces of water into a 2-cup measuring cup. Add 5 to 10 drops, about 1 teaspoon of bromothymol blue. Stir until the solution turns blue. Observe the color change. So that's when we'll look at the pH and see what it is. This third step, create a carbon dioxide rich solution. Gently, so you don't bubble it out. You don't want to splash it all over everything. Gently blow into the bromothymol blue solution using a straw for about 30 seconds. This adds carbon dioxide, making the solution greenish yellow. Remember, bromothymol blue is an indicator that turns yellow in the presence of CO2. Putting CO2 is going to make it turn yellow. Step 4, set up control and experimental groups. Pour the carbon dioxide rich bromothymol blue solution into 3 test tubes or jars. Label 1 as control. So I've got mine right here. I just wrote control on the side of my tube. Label 1 as experimental light, EXP light, and one as experimental dark, EXP dark is what I put. When you guys look at that, is it right side up or is the wording backwards? I'm curious. Can you see the EXP dark?
Yeah.
Okay, I wasn't sure. Sometimes it's reverse. All right. Then step 5, experimental setup. We're going to put a plant in both of the experimental test tubes and seal them with a lid. We're going to leave the control without a plant and seal it with a lid. Then we're going to wrap the experimental dark in foil to block out the light and put all of them under a light source. We are going to use this chart, and over the next 24 hours at 15 minutes, 30 minutes, 1 hour, 8 hours, and 24 hours, we're going to observe the color. Now, for those of you who are not doing the experiment yourself, I will send you a picture, some pictures this time tomorrow. And those pictures is what you will use to fill out this chart and to answer the analysis question. So if you look at the last page of your lab sheet, the analysis is what you're going to finish as well. And we'll talk about that next week. But you'll use the pictures that I send you tomorrow in order to do that. If you're doing it yourself, you'll look at your own. Okay? Now we're going to go back and actually do the lab. So you're going to need to look at your lab sheet because I'm going to stop the share and I'm going to pin mine so it'll be bigger. And I'm going to tip this down so you guys can see what I'm doing here. Okay? All right. So let me move all this so you can see it a little bit better. Okay, so I'm going to put on my goggles. That's step 1. Now I'm going to prepare my bromothymol blue solution. I have mine in a flask right here. It's just clear water in this flask. And I measured out 150 milliliters right there on the side. Now, the next thing I'm going to do is add 5 to 10 drops of bromothymol blue. I want to put in enough drops that I get a nice blue color. I might have to put in more. So if you're doing it yourself at home, you want to get a color that is, I'll show you about what the color. So the first one that I put in, I'm going to swirl it. Did that change it? Let me get my straw to stir. Didn't really change it. Maybe a tiny bit. Maybe, well, see, that's reflecting. That's showing these blue lids right there. All right, let me put in all the rest. 3, 4, 5, 6, 7, 8, 9, 10. I've got 10 drops. Let's see if I need more. I've got a nice blue color there. Probably not going to get much more than that blue, but take a look at that one. So that one's going to be right around the like 7.5 or 8 pH, because that's what we looked at on that pH scale. Let me try to add just a couple more, see if we can get it. It'll get to the point where it just really won't go much bluer. I think that's probably it. Yeah, that's not changing it anymore. So I'm going to put away my bromothymol blue. All right. The next step is to add the carbon dioxide. I want to create a carbon dioxide rich solution. This doesn't have it doesn't have much CO2 in it now. I can tell because it's blue. When I add the CO2, it's going to turn a yellow or green color. So I'm going to gently blow into this until it starts turning yellowish green. All right, so you guys can watch as it changes color. It's turning a little bit paler, a little paler blue, and you can see a little green tinge to it now, but there's not enough. I got to keep adding CO2. Okay, that's a pretty significant difference now. That's pretty pale green. Let's try one more. I think some of this blue that you guys are seeing is reflecting off my shirt because I've got that. So let me put that in front of that. Yeah, there you can really see. If I block that, we really changed the color. It's this pale yellowish green color right there. All right, so I'm now to step 4. I've made my carbon dioxide rich solution. Now I'm going to set up my control and experimental groups. So I'm going to pour the carbon dioxide rich bromothymol blue into these 3 test tubes. The control, the one labeled experimental light, and the one labeled experimental dark. All right, so let me pour those in. I
May
don't want it all the way to the top because I want to have room to put the plant in. What, Bethany?
How much are we supposed to put in each one?
So you want your jar to be fairly full, but you want to have enough room right now for the plant. So fill it up to, about a half an inch or so from the top. If your jar is really big, your little jars that you're using, you can make more of this bromothymol blue solution. Same way, blow the bubbles into it or the carbon dioxide into it to fill them up so they're pretty full. Okay, so I'm just going to I'm not going to fill them all the way to the top. I'll do that after I put the plant in. All right. Now, step 5. I am going to place a plant in both of these 2 experimental ones. The one that is a control, I'm going to leave that without a plant. So I want to put a plant down in the experimental light. And I want to put a plant down into the experimental dark. The plants are approximately the same size that I'm putting in both because I want to eliminate as many variables as I can. Now, I got a little bit on my fingers when I poked it down in there, so I'm going to use my wet paper towel to wipe my hands off to make sure that like I said, it doesn't burn you, but it is a little bit of a skin irritant, so you don't want to leave that on there. All right. Now I've got a little bit left in here. I'm going to top these off so that they all have about the same amount of the bromothymol blue solution. And I have enough to pretty much fill up my test tubes. There's still even a little bit left. Now I'm going to put the lids on. I'll put those lids on nice and tight so it doesn't leak. There is my experimental dark. Here is my experimental light. And here is my control. All right. Now, I'm going to note the initial color of the bromothymol blue in each of these tubes. So you can see it's that pale. Let me hold up my thing again so you can see the color. It's that real pale. It's almost clear. Got just ever so slight of a yellowish green color in it. But that's really pale. I blew more carbon dioxide than usual today. All right, so they all have the same color. So on your table, under initial, you're going to write the color. Pale greenish yellow or yellowish green for all 3, because that's what they are. Now, we will not still be here 15 minutes from now. So 15 minutes from now, you'll have to look at them. If you're doing it yourself, you'll look at them yourself. If not, I will send all of the results tomorrow and you will use those results. All right, so the last thing we're going to do is look at the one that says experimental dark. I'm going to wrap it in foil in order to block out all the light. Now, every time I want to evaluate them at the intervals, the 15 minutes, 30 minutes, I'm going to unroll it so that I can look at it and then just roll it back up. So an hour from now, I will open it up like this. I will take a look at it, evaluate the results, and then I'll roll it back up. And I'll do that every time I want to fill out my chart. Okay, that's the lab. That's the setup. These 3 now will be placed under a light source. So I'm going to tip my camera back up. Here we go. I'm going to take my goggles off because I'm done. And I will share my screen with you guys again. All right, so we are going to, you're going to be filling this out, but I have a couple questions for you that we're going to talk about while we're, theoretically waiting for the 24 hours to go by. So once again, right here, you're going to have yellowish green, yellowish green, yellowish green, because that's what's the color that it started out. It's a real pale yellowish green color. At 15 minutes, I'll look at it again. I'll take a picture, and then half an hour from now, I'll take a picture, an hour, 8 hours, and 24 hours. So about 24 hours from now, I'll send you guys all of the pictures. so you can use them. And you will use them, you'll fill out the chart, and then you'll use that information to answer the questions that are in the analysis part of the lab sheet, that very last page. That's what we'll talk about next week. So here's a couple things to think about. What color was the original bromothymol blue in water before I blew into it?
Blue.
Blue. Yeah, it was that very nice bright blue color. We can look back here at the pH. Probably was about 7.5. I don't think we had this dark right here. We had this one, about a 7.5. All right, great. So the next question then is, why did I put on goggles?
To prevent the bromothymol blue from burning your eyes.
Yep, exactly.
Okay.
Yep, exactly. Safety first. We just always want to be safe. So if you got any of it on your skin, use your paper towel to wipe it off. If you got a lot on your skin, go wash your hands. Go wash it with some soap wherever you got it. All right. Why did I make that control test tube? The one that I didn't put a plant in. What was the point of that?
To see the
That's-
reaction if. To see the
That's-
react.
Go ahead, Luke.
To see the reaction without a plant.
Yeah, we want to have something to compare it to. Because if tomorrow my experimental light and my control are identical to one another, if they both turn a different color or neither one of them does, then I can't say for sure that the plant is what made the difference. There would be some other variable at work right there. I have to have something to compare it to. All right. Why did the color change to greenish yellow when I blew into it? Maria.
Because the bromothymol blue was reaction with the carbon dioxide.
Exactly. It was indicating the presence of carbon dioxide. So bromothymol blue turns that yellowish green when there's carbon dioxide in the solution. That's why the color changed. Why am I using a light source? Oh, speaking of the light source. So if you still have daylight at your house, put all three of these in a window where they can be exposed to daylight. Then once it starts getting dark, then you're going to need to move them into a place where you can leave a light on overnight. We need to have 24 hours of light on these in order for this to work. So just like a bathroom or something, a place that you can leave a light on overnight works really well. And if you're doing it yourself, I do know that at the 8-hour mark, you'll probably be sleeping. So just do it and, keep track of that before you go to sleep. So if it's only been 6 hours, that's fine. Just look at it again when it's been 24. Okay, so why did I use a light source? Why am I telling you to put this in the light overnight?
Because that's how photosynthesis occurs.
Yeah,
It's
because
just like-
the light reaction requires light. The whole process of photosynthesis can't happen unless it's under a light source. Now, when I say comes off, I mean is a product. What comes off a plant during photosynthesis? What is the product that comes out of the plant? Maria.
Oxygen.
Oxygen. Now the plant also makes glucose, but the glucose doesn't come out. The glucose is stored in the plant. The oxygen is released. It's a waste product. And what color is bromothymol blue when there's oxygen present and low levels of carbon dioxide? Bethany.
I wasn't raising my hand.
Oh, I'm sorry. Looked like it. Yeah. So what was it when I first mixed the water with the bromothymol blue before I blew into it? When there
Blue.
was oxygen. Yeah, that's because there was oxygen and no CO2. So it was blue. So the bromothymol blue is blue when there's oxygen and low levels of carbon dioxide. So let's hypothesize now. What color do you expect to see in this control at the end of the experiment? What do you think is going to happen to the control bottle, the control test tube? Bethany.
I don't think it'll change that much.
It shouldn't. If it changes, it's because there's, it's exposed to something. It could be because there's contaminant in the tube, or there's some carbon dioxide that is getting, or oxygen that's getting in. There could be a leak in my tube. That's a possibility. But we expect it to be the same. We expect it not to change at all. What about the one that has the plant but's wrapped in foil? What color do I expect to see in this tube? What do you think?
Blue.
Yeah. No, I'm sorry. No, I expect it not to change because it can't go through photosynthesis. Right? Does that make sense? I'm preventing light from getting at it, and the photosynthesis requires light. So it should be the same color as the control because neither one of them will be performing photosynthesis. All right. And then finally, what color do I expect to see in the tube that contains the plant and was exposed to the light source? What do I
Turn
expect?
blue.
Yeah. Why?
Because it was exposed to light, which made it go through photosynthesis,
Yeah.
which exposed it to, I can't remember, which made it turn blue.
Yes. So what's going to happen? There's a lot of carbon dioxide in this now. What does a plant need to do photosynthesis? Water and carbon dioxide. It's down in the water. That's what's going to get the water. Where's it going to get the carbon dioxide that it needs? From the carbon dioxide I put in. From the carbon dioxide from my breath. That's where it gets the carbon dioxide that it needs. When it's in the presence of carbon dioxide, bromethymol blues yellow. Once I start taking that carbon dioxide out in the process of photosynthesis, now it's going to turn back to blue because there won't be carbon dioxide in there to turn it yellow. There will be oxygen. When it's in the presence of oxygen, it's blue. When it's in the presence of CO2, it's that yellowish green color. All right, so you're going to look at your experiment 8 hours later or before you go to bed, and then 24 hours later, and make sure that you complete your chart. If you did the lab yourself, use your results. If you're looking at mine, I'll send them tomorrow. You do not have to turn this in, but we're going to talk about it next week. So I do expect everybody to have your charts completed, whether you did the experiment or just observed here. Have your chart completed and fill in all of those analysis questions. All right. And then I did have a request here for a couple of photosynthesis jokes. So we will end with these. What does the little plant say when mom gives him dinner? Oh, mom, sunlight again. And just the steak for me, and my friend will have the carbon dioxide in the water with the sunlight. And then for the musicians among you, a photosynthesizer. I like this one too. One tree talking to the other. Can you believe that people inhale the gases we expel? Sick, right? Yeah, think about it. We are literally breathing in the waste products of plants, but it's a good thing we have them. All right. We will continue the discussion of photosynthesis, talk a little bit about cellular respiration next week. So make sure that you bring your charts back with you and your analysis questions. Anybody have any questions? All right, terrific. Have a great week. Thanks for coming, everybody.
You have a nice day.
Thank you.
Bye. Thank you. Have a nice day.
You too.